A user on Reddit just posted an interesting story detailing that from the late 2000s to mid-2010, they worked as a molecular biologist for a national security contractor in a program to study Exo Biospheric organisms. They posted the following on Reddit, but oddly enough, after only a few hours, their messages were deleted and their account was taken down. They said the following: "From the late 2000s to mid-2010, I worked as a molecular biologist for a national security contractor in a program to study Exo Biospheric organisms. The aim of the program was to elucidate the genome and proteome basis of these organisms. Although the study of OBCs has been going on for decades in other programs, the new high-throughput DNA sequencing technologies of the late 90s unblocked stagnant research in this area. Since then, several breakthroughs have led to significant advances in our understanding of the genome and proteome of these beings. What we've learned so far has enabled us to outline some disconcerting perspectives about our place in this universe. Briefly, we've discovered that the Exo Biospheric Organisms genome is a chimera of genomes from our biosphere and from an unknown one. They are artificial, ephemeral, and disposable organisms created for a purpose that still partially eludes us. I'll be substantiating my statements after a brief introduction. The reason for disclosing these secrets is quite simple. I believe that every human being has the right to know the truth, and that to progress, humanity needs to divest itself of certain institutions and organizations that will probably not survive these revelations in the long term. I'm aware that I'll have very little impact in this regard, but I still believe that small leaks are necessary to break the dam of misinformation on this subject. When the governments will eventually reveal these secrets, there will undoubtedly be a societal upheaval, but in my opinion, the longer we wait, the worse it will be. I choose to divulge what I know anonymously out of selfishness for the well-being of myself and my family. I'm aware that this diminishes the reach and credibility of my message, but it's the furthest I am willing to go. I chose this forum because it offers a good compromise between anonymity and popularity. In order to protect my anonymity, I will be purposely vague or even contradictory about any information that could identify me. I'll even introduce red herrings in this respect. I want to make it clear that any information related to the subject of the research will not be treated in this way. Before going any further, please excuse me if you find it difficult to understand what I'm explaining. Some parts of my text are very technical. It's difficult to find the right balance between vulgarization and scientific explanation. I'll continue by talking about myself. I simply want to introduce a perspective on the type of people who work there. I have a PhD in molecular biology. I didn't actively seek to be part of this program. Rather, it was a stroke of luck that introduced me to one of the senior scientists. I met this person at a conference where I was presenting a poster on my PhD research. When I think back, I don't believe he was impressed by what I was presenting, because it was quite frankly a project that wasn't going anywhere. I think it was rather the most important aspect of a professional life, the attitude and the ease with which you make connections. Shortly afterwards, I graduated and received a call from this person offering me a position. At the time, everything pointed to me working in a regular laboratory. I did a series of 3 increasingly suspicious interviews, each in a different location, where my scientific background and knowledge became less and less relevant. The first was with 2 of the senior scientists, the second and third with people I've never seen again, and who were obviously not interested in science. Sometime after the interview, I was asked to go to a 4th location, where what seemed like a corporate lawyer presented me with an NDA. He made sure not only to explain every detail, but also that I understood the consequence of not respecting it. The first employment weeks were by far the most memorable, although I spent most of that time in an archive room. It consists almost exclusively of reading about the subject of study and to get us up to speed. There's no secret Wikipedia or even a reference book to guide us. There are only dry reports, memos, presentations, procedures, and SOPs. These documents are almost exclusively about the biology of Exo Biospheric organisms, but there are also a few that deal with other subjects such as their food, religion, or culture. There were no documents on their technology. As mentioned above, the aim of the project is to gain a better understanding of the EBO genome and proteome. To achieve this, a team of around 20 scientists 4 senior scientists and a director was involved. The scientists, like myself, had as their main responsibility to carry out the technical work. As each scientist had, to my knowledge, a PhD, we were all somewhat overqualified for what is ultimately a technician's job. The senior scientists, who make full use of their diplomas, had the task of designing the assays and had a supervisory responsibility. They were also in charge of training new employees and sometimes even came in to do technical work. The director, of course, was the person in charge who dictated priorities to the senior scientists. He was rarely on site, and the few times he was, it was to attend meetings. Other than the scientific staff, there were security guards working for one subcontractor or another. There were no support staff, such as janitors or maintenance workers. Scientists were responsible for this kind of work. In addition, logistical constraints ensure that every scientist is capable of carrying out any technical activity. The laboratory itself is located in Fort Detrick, Maryland, in a building used for legitimate biomedical research. The clandestine operations are carried out in a restricted part of the basement, out of sight from regular workers. Contrary to what one might imagine, the biosafety level is not maximal for this type of research. Indeed, the lab containing Exo Biospheric Organism samples, or derived cell cultures, is biosafety level 3, while the lab where assays are conducted are only biosafety level 2. The biosafety level 3 area of the facility includes a freezer room and a cell culture lab, and is only accessible through an antechamber from the biosafety level 2 section. Exo Biospheric organism carcasses are preserved in horizontal freezers at a temperature of -80 degrees. To maximize the preservation of these organisms, they are preserved in vacuum bags, and the air in the room is controlled to minimize humidity. There are only 4 entities, and none of them are complete. It's obvious that these creatures passed away as a result of major trauma. It is acknowledged that there are more exobiospheric organisms at other locations. The cell culture laboratory, as its name suggests, is where cell lines derived from exobiospheric organisms are grown and related activities are performed. I'll talk in more detail about these specific cell lines later on. The biosafety level 2 part is mainly used for assays, immunohistochemistry, genetic engineering, immunocytochemistry, and storage. There's also a cell culture lab, but this is used for more traditional cell lines. Other than the labs, there are all the amenities you could find in an office. Note that the internet access is limited to senior staff and up. There is, however, an intranet for bioinformatics needs. On the subject of the biology of these beings, I'll start by discussing genetics, then their gross anatomy, and finally, their biological systems. For the sake of clarity, the information that I provide here is an aggregation of what I have observed and what I have read. I will make many comparisons with human anatomy because it is the most logical reference. First, I'd like to discuss their genetics. Their genetics are like ours, based on DNA. This fact was very puzzling for me when I first learned about it. We imagine that beings from an alternate biosphere would have genetics based on a completely foreign biochemical system, and surprisingly, this is not the case. Several conclusions can be drawn from this surprising revelation. The one that immediately comes to mind is that our biosphere and theirs share a common ancestry. They're eukaryotes, which means their cells have nuclei containing genetic material, which suggests that their biosphere would have been separated from ours sometime after the appearance of this type of organism. The term exobiospheric organism is actually a misnomer, but as it's a historical term, it's still used. Their genetics are not only based on the same genetic system, but they're also even compatible with our own cellular machinery. This means that you can take a human gene and insert it into an exobiospheric organism cell, and that gene will be translated into protein. And this, of course, works in reverse with a human gene inserted into an exobiospheric organism cell. There are important differences in post-translational modifications that will make the final protein non-functional, but I'll discuss these later. Their genome consists of 16 circular chromosomes. You're probably familiar with the concept of Introgenic region, or junk DNA. These are basically DNA sequences that don't code for proteins. These are evolutionary residues, transposons, inactivated genes, and so on. To give you an idea, in humans, Introgenic regions represent approximately 99% of our genome. I'm aware that these sequences aren't completely useless. They can be used as histone anchors, as buffers to protect coding DNA from radiation, or even as alternative open reading frames, but that's rather peripheral. What's particularly striking about the Exo Biospheric Organisms genome is the uniformity of these intergenic regions. We see the same sequences repeated everywhere, and the distance in BP between the genes is virtually the same throughout their genome. The result is a minimalist, highly condensed genome. In fact, it's much smaller than ours. Moreover, the quantity of protein-coding genes is even significantly lower than ours, probably due to genetic refinement, but also to biological processes that are absent in exobiospheric organisms. The uniformity of these sequences is a major indication of the artificiality of these beings. There is no complex organism on Earth that has such elegance in its sequences. There is no evolutionary pressure that can lead to this kind of characteristic other than genetic engineering. Speaking of genetic engineering, following sequencing of their genomes, we noticed a troubling and universal characteristic in the 5 of the regulatory sequence of each gene, which we call the Tripalindromic region. The Tripalindromic region are 134 sequences containing, as its name suggests, 3 palindromes. In genetics, a palindrome is a DNA sequence that, when read in the same direction, gives the same sequence on both DNA strands. They serve both as a flag and as a binding site for proteins. The 3 palindromes in the Tripalindromic region are distinct from one another and have been poetically named 5 Tripalindromic region, M Tripalindromic region, and 3 Tripalindromic region. The chromosomal address is composed of 4 BP and is identical in each Tripalindromic region of the same chromosome, but distinct between each of the 16 chromosomes of the genome. The gene address is a 64 BP sequence that is unique for each gene in the whole genome. It's therefore understandable that the Tripalindromic region serves as a unique address, not only for numerically identifying a gene, but also for identifying its chromosomal location. For those with only a basic knowledge of genetics, this is completely unheard of. No living thing in our biosphere has this kind of precise address in its genome. Once again, the presence of Tripalindromic region cannot be explained by evolutionary pressure, but only by genetic engineering on a genomic scale. Tripalindromic region opens the door to several possibilities. One of them suggests that exobiospheric organism geneticists can insert or remove a gene from a cell in a way that is far more targeted and efficient than our technology allows. No proteins have been identified in the exobiospheric organism genome that interacts with the Tripalindromic region. Rather, we believe that these proteins are exclusively targeted by external genetic engineering tools, probably used at the zygotic stage of embryonic development. The nature of these tools is unclear, but we definitely don't have anything like them. The probable absence of these proteins from the genome is a further indication of their artificiality. Given the high probability of artificiality of their genome and the apparent ease of modifying it with biomolecular tools, it's not out of the question that there could be polymorphism between individuals depending on their role and function. In other words, an individual could be genetically designed to have characteristics that give it an advantage in performing a given task, like soldier ants and worker ants in an anthill. Note that these previous statements are speculation. To my knowledge, only one individual genome has been sequenced. I can't make a definitive statement on genetic variation between individuals. I've talked a lot about intergenic regions. Now I'll briefly discuss introgenic sequences. Briefly, because there's not a lot less to say despite its obvious importance. Much like ours, their genes have silencers, enhancers, promoters, 5 UTRs, exons, introns, and 3 UTRs. There are many genes analogous to ours, which is not surprising given the compatibility of our cellular machinery. What's disturbing is that some genes correspond directly, nucleotide by nucleotide, with known human genes or even some animal genes. For these genes, there doesn't seem to be any artificial refinement, but rather a crude copying and pasting. Why they do it is nebulous and still subject to conjecture. There are also many genes which are not found in our biosphere, whose role has not been identified. Finding the purpose of these novel genes is one of the aims of the program. I'd like to note before going any further that this heterogeneity of genes of known and unknown origin is an undeniable proof of the artificiality of Exo Biospheric organisms. To conclude with genetics, the mitochondrial genome at the time I was working there had not yet been sequenced. It's safe to assume that this genome would also be streamlined and possibly has some version of Tripalindromic region. Transcription and translation and protein expression. I briefly introduced the differences in post-translational modifications between human and Exo Biospheric organism. This is hardly a surprise, as we often see the same thing between different terrestrial species. Obtaining a viable protein from a DNA sequence is a complex process involving hundreds of protein intermediates, each with a precise and essential role. A minor variation in this assembly line can lead to functional irregularities in the final product. So, it's no surprise that there are setbacks along the way when the first Exo Biospheric organism gene transfection attempts failed to produce the desired functional protein in human cell lines. Fortunately for us, the work of what I imagine to be another team at another site has led to the development of an Exo Biospheric organism cell line named EPIG-11, derived from epithelial tissues. With this tool in our hands, we were able to transfect and overexpress proteins of interest in order to eventually purify and study them. For your information, we use a biological ballistics delivery system for our transfection needs because other methods are not very effective with cells of this line. For example, the viral vectors tested cannot be internalized by EPIG-11 and lipofection is too lethal. EPIG-11, like most eukaryotic cell lines, enters a phase of exponential growth when exposed to fetal bovine serum. It's only half surprising that a cell line from such an exotic source should be sensitive to the growth factors present in FBS. In my opinion, this can be explained by the addition of animal genes to the genome, such as growth receptors. Gross anatomy. They are morphologically very similar to the grey aliens that are part of modern folklore. Their height is about 150 centimeters. They have 2 arms, 2 legs, and a head. Still, there are some notable differences. The grey skin that is often described in folklore is in fact a biosynthetic film which, likely, serves to protect the Exo Biospheric organism from a hostile environment. It doesn't provide effective protection against temperature changes, but it does offer adequate protection against the passage of liquids. It's possible that this film confers other advantages, but my knowledge on the subject is limited. Under the grey film, the epidermis is rather white, and the texture is very regular and without any hair. We do not see any defect other than the folds near the joints. It's described as greasy in one report, but that's not something I've observed. The same report states that a strong, lingering smell of burnt hair and ammonia is present when the film is removed. There are a lot of pores on the skin, crossing from the epidermis to a gland in the hypodermis. These glands and pores are the terminal part of the excretory sudoriferous system, which could explain the previously mentioned smell. The head contains 2 large, oversized eyes, 2 nostrils without protuberance, a narrow mouth without lips, and 2 ear canals without auricles. There is a mandible, but the musculature is vestigial. There are no teeth or tongue in the oral cavity. The nasal cavity where the nostrils meet is compact, and does not rise cranially, but extends axially. There appears to be no equivalent to the olfactory bulb in the nasal cavity. The mouth leads directly to the esophagus, and the nasal cavity to the trachea. The trachea and esophagus do not communicate. Like the skin, the eyes are covered with a semi-transparent biosynthetic film that offers the same environmental protection while providing protection against certain wavelengths and light intensity. When the film is removed, a more traditional eye is revealed. It's about 3 times larger than a human eye, and there are no eyelids. The size of their eyes suggests they have excellent night vision. It seems paradoxical to cover them with a semi-opaque film. Perhaps they only need to wear it in a bright environment. Their sclera is the same color as their skin, the iris is pale grey, and the pupil is black and oversized. The lens is rounder than a human, and the musculature used to adjust focus is more developed. On the retina, there are at least 6 types of cone cells. The responsiveness of each of these 6 types of cone is specific to a wavelength band, with a minimum of overlap between each other. The result is a broader visible spectrum. As mentioned, The outer ear has no auricle, and the ear canal is unremarkable. The inner ear has all the characteristics of a typical vestibular and cochlear system, although the curvature of the cochlea is more pronounced than a human. This probably results in greater hearing acuity for low frequencies. The brain is tetraspheric and is composed of 4 major sections. The sections are separated by transverse and longitudinal fissures and are connected to the central lobe, which acts as brainstem and cerebellum. The volume of the brain is around 20% superior to that of a man of the same height. It has a much more pronounced level of gyrification than an average human. Moreover, the ratio of glial cells to neurons is also slightly higher than in humans. It is important to mention the presence of nodules on the central lobe. Histological analysis of these structures reveals a kind of intricate biological circuitry. It is speculated that these nodules are essential to interact with their technology. Consequently, determining the proteome of these structures is an absolute priority for the program. The neck is proportionally longer than that of a human, and at the same time relatively thin. As mentioned, the esophagus and trachea are separate. There are no vocal cords in this region. The musculature of the thorax is underdeveloped. Muscles equivalent to the pectoralis major can be seen. We can also see the trapezius and deltoid muscles. The sternocleidomastoids are well defined. The ribs and sternum are clearly visible. The abdomen is wider than the thorax and bulges slightly forward. There is no navel. Their hands have 4 digits, including an opposable thumb on the medial side. They have no nails, and the texture of their fingerprints is composed of concentric circles. Fingers are proportionally much longer than in humans. Unlike humans, finger musculature is entirely intrinsic to the hand. In other words, the muscles used to move the fingers are not in the forearms, but entirely located in the hands. At first glance, the feet consist of just 2 digits, but a necropsy soon determined that each toe was made of 2 fused digits. The medial toe is marginally longer than the distal toe. The feet are relatively longer and narrower than in a human. Their musculature, however, is vestigial. The Exo Biospheric Organisms' endoskeleton is very similar to ours, at least in terms of composition. There's collagen, hydroxyapatite, but also copper oxide crystals where marrow would normally be found. The blood cells of the myeloid lineage therefore mature in a different location than in humans. A transverse section of the bone reveals osteon and osteocytes. There appear to be few osteoblasts and no osteoclasts. This indicates that the bones are no longer growing and cannot absorb the minerals present or adapt mechanically to changes in posture. Their cellular respiration is equivalent to ours. They need to oxidize organic components to produce energy. Their lungs have no reciprocating action, but rather have a unidirectional flow of air, similar to those seen in birds, which is more efficient than ours. It is speculated that this is in response to the brain's elevated metabolic needs. Vocalization is produced by vibration of the wall membrane at the junction between the 2 air sacs. The circulatory system of exobiospheric organisms is rather analogous to ours. The heart is located in the mediastinum, but in a more medial position, directly beneath the sternum. The heart has 2 ventricles and 2 atria. There is an aorta, a pulmonary vein, a pulmonary artery, and a vena cava. Blood flowing to the pulmonary capillaries via the pulmonary artery is pumped against the flow of air, maximizing gas exchange efficiency. The blood-gas barrier is relatively narrow in these capillaries, at least compared to a human. Then, oxygen-rich blood is returned to the heart and then expelled into the aorta and the rest of the body. Before returning to the heart, the blood will pass through the hepatorenal organ, which, among other things, filters and controls osmotic pressure of the blood. The blood itself is also analogous to that of a human. However, the proportion of plasma is much higher. Albumin is in similar proportion, hormone levels are much lower, metal ion levels are much higher, and glucose levels are significantly higher. The color of the blood is brownish, given the higher proportion of plasma and concentration of metal ions. On the cellular side, there are erythrocytes which, in addition to hemoglobin for binding oxygen, display several complexes capable of binding copper ions. It's not clear what role these copper ions play, but we believe it neutralizes blood ammonia, among other things. Several cell types with leukocyte characteristics have been observed, but no comprehensive knowledge of them exists. So, what do you make of this interesting story? Be sure to leave your questions and answers in the comment section below and help us to grow this community while working to solve these unexplained mysteries. Thank you for watching, and don't forget to subscribe for more videos.